Ann Summers is a professor of microbiology at the University of Georgia in Athens, Georgia. She studies bacterial metabolism of mercury, the biochemical basis for cellular damage by and recovery from inorganic and organic mercury compounds, and the mechanism of spread of antibiotic multi-resistance in bacteria. With degrees in chemistry, microbiology from the University of Illinois, and in molecular biology from Washington University at St. Louis, she is a fellow of the American Academy of Microbiology, of the American Association for the Advancement of Science, and of the IAOMT. Her work has been funded by NIH, NSF, DOE, EPA, and several private sources, including the J.S. Guggenheim Foundation and the IAOMT. Please give a welcome to Ann Summers. Thanks.
Thank you. Thank you. Yes. Let me read the AGD disclaimer first of all. I do not have any financial interest of a product in my talk or any companies offering grant monies for this continuing dental and medical education program. So thank you very much for inviting me to speak at this meeting. It's always a pleasure to get together with this group because it's an exciting group intellectually and personally to see the dedication of so many people who are concerned about this and educating themselves in the risks associated with dental materials and mercury in particular. So I have worked in a couple of different areas over the years. I guess I first came into this particular aspect of mercury biology some almost 20 years after I first got interested in mercury as a subject of study. And I will tell you about the studies that I have done in the context of amalgam mercury. There are many other areas of concern for mercury, and from our funding sources, our major funding source on this work has been in recent years from the Department of Energy, though we have been funded by NIH and NSF as well in prior years. But the Department of Energy has a huge mercury problem, particularly at Oak Ridge National Laboratory. And they are concerned with cleaning that up and also understanding the microbiological implications in stewardship of that area. So without further on that, after Mark's talk, where I know he was talking about mathematics and other kinds of technical issues, I'm going to say, hold on, we're going to talk about molecular biology, and I will leave some time. I hope I will be able to do that, leave some time at some point to answer questions that may come up. The talk's really divided into a couple of sections. So the first section does have to do with our beginning studies that had to do with antibiotic resistance and association with exposure to mercury. And so that'll be the first part, studies in living animals. And then I'll segue over into our more recent studies that are at a cellular and biochemical level. And all of these studies have been done using bacteria. We have studies in monkeys where the living animal hosts for the bacteria. And so I've titled this, What Bacteria Have Told Us About Mercury Intoxication. So this will be associated with the antibiotic resistance story, and then the second section is more associated with what are the most vulnerable targets of damage of mercury, by mercury in the cell. Because every disease manifestations, the various kinds of tissue damage that we've talked about this afternoon and other kinds of syndromes that are associated with any kind of intoxication have at their base some misfunction at the cellular level. Something's gone wrong in these amazingly complex and sophisticated little globules that constitute the cells of our bodies. And we have two purposes in doing work at the cellular level. One of them is actually to find out why and how mercury is toxic. You may not know that mercury is a very paradoxical toxin, but I'll point that out later as we come along. It's paradoxical in many ways. And we want to get to the bottom of that and figure out what our most vulnerable targets in our cells are. And just for the basic science aspects to figure that out, but also for the utility that can have to know those things and what kind of contribution knowing that can make to larger areas of human biology and health. So I will come to those as we go along. And first of all, let's talk about microbiology. Everybody in this room is walking around with about a pound or two of a community of microorganisms in their GI tract. There are also microorganisms in other places. They're in the upper, respiratory tract, and down even into the stomach. And ideally, the liver is not colonized, but it can be, in the gallbladder and so forth. This is the small bowel, which in most cases is not colonized at its upper end, but it can be colonized nearer to the lower end. But the major community that we have in our body that is in the large bowel, in the colon. And there are literally more cells of bacteria in us and on us than there are cells of our own body. Okay, we're outnumbered. We are walking communities, we're walking ecosystems, and the larger area of microbiology has only come recently to an appreciation of this particular reality. So let's talk a little bit from a very narrow perspective, our Mercury perspective, about what this is all about. 90 to 95% of the mercury that comes into the body orally, mercury of any kind, whether it's fish or whether it's amalgam mercury or whether it's other kinds of exposure, 90 to 95% of that is eliminated through the GI tract. Okay, some of it goes through portal circulation and does go out through the kidneys. All right, but given that and the robust and diverse microbial community in the lower bowel, which is capable of taking ionic mercury to mercury vapor, mercury zero, and also to methylating mercury, both of which readily cross the membranes of epithelial cells and go back into circulation. In other words, microbes and bacteria primarily in the GI tract mediate an enterohepatic cycle for mercury. So, whereas most of it can go out this way and most of it appears to go out, what we see coming out is not necessarily all that went into the GI tract because of this enterohepatic cycle. It can go out through the epithelium before it actually exits through the anus, courtesy of being volatilized by the microbial community that lives in the GI tract. Another interesting fact is that 80% of the body's immune cells line the GI tract from the mouth to the anus. Okay? The gut-associated or mucosal-associated lymphoid tissue, GALT or MALT, lines the GI tract. Okay? It's our first native contact with exogenous ligands from the outside. Okay, food is one of them, and early in our lives we develop a tolerance for foods so that we can eat them. But everything that passes through from the beginning is certainly a potential allergen or is in position to intoxicate the cells that are kind of sorting out what is an allergen and what isn't an allergen from our environment. Okay? And helping those cells maybe not do such a good job. So 80% of the ground cells, the basic cells with which we contact the external world in terms of immune function are in the GI tract. Interestingly, about 80% of the serotonin receptors in our bodies, they're all in my brain, right? No, 80% of them are in the GI tract. In fact, the gastrointestinal system, it's not shown in this, has such a large wad of neuronal tissue surrounding it that it's been referred to as the brain in the gut. Okay? So there's a chunk of neuronal, highly innervated tissue surrounding the GI tract and subject to communication, chemical communication with it, that's nearly the size of the spinal cord. Okay, and we know very little about what it does, but it does communicate with the gut contents, including the bacteria. So this is the gut having its own brain. Okay? So this is the context in which we were concerned to look at bacterial relationships to mercury. Let's see, let's punch that thing. Now, before we came to study the subject of amalgam-related mercury, we knew about bacterial mercury resistance. I won't go into the details of why that was, but we had learned, not just me, but scientists in general, had learned that many eubacteria, which is another word for just plain bacteria, this is a little square, a little oblong bacteria. Bacteria have chromosomes. They're circular DNA, unlike ours, which are linear. And this is where the housekeeping genes, the genes to be a cell and know how to divide, know how to metabolize, exist. But bacteria can also have these small supernumerary chromosomes that are called plasmids. They're made out of DNA, and they have functions that are adventitious in certain settings. And what's cool about them is that the bacteria can exchange these things. They don't usually exchange their whole chromosomes, but they can exchange these little plasmids out there in the wild, here in the wild. Okay, they can be doing that all the time. All right? And what one frequently finds associated with plasmids are genes that are unique to defending yourself in or taking advantage of a particular ecological niche in which you find yourself. Plasmid-encoded genes, for example, are important in the bacteria that live near leaking gasoline tanks in gasoline stations. They're helping the bugs that live in the soil there metabolize octane. Okay, yum, if you've got the genes on the plasmid. In us, plasmids are more often associated with antibiotic resistance. Okay, now this is a plasmid, and this is a section of that plasmid that itself is also mobile. It's called a transposon. So not only can the plasmid move from one cell to another, but the transposon itself can move from the plasmid to the chromosome. So there's several different levels of gene mobility in bacteria. But this one in particular, called transposon TN21, carries several different antibiotic resistance genes here, to sulfadiazine, to things like aminoglycosides and tetracyclines and so forth. And that particular batch of multiple antibiotic genes, antibiotic resistance genes, happens to be right next to the genes for mercury resistance, which we were studying for a long time anyway. And so I'm not going to go into detail here in talking about these. I'll come to that a little bit later. But the suite of proteins is capable of converting ionic mercury into mercury vapor, which I just mentioned. That's something that bugs can do, monoatomic mercury vapor, which is so permeant to our epithelial membranes. It also can, there are other genes, a part of this whole story, that can degrade methylmercury. But I'll come back to this a little bit later in the talk. So we were studying this anyway, and we knew that this was the case, and we knew that this was the case, but we didn't really, that was not something we were interested in studying until we had the opportunity to look at some human samples from a study that we did with Stuart Levy at Tufts University. And in that study, we were looking at fecal samples from people living in Boston because there was a great interest in just how much antibiotic resistance there was out there. And these were studies that were begun actually in the late 70s and concluded by us initially in the early 80s. And what we found, looking at individual human fecal samples, streaking them out on a Petri plate, letting them grow up, and then assaying them for their resistances to various things by a fairly simple microbiological procedure called replica plating. So we could find hundreds of colonies on the plate, and by replica plating on different media, we could find out, is it resistant to TET? Is it resistant to sulfa? Is it resistant to mercury? And so we did this, and what we found among all the subjects that we looked at, and we looked at about 650 human subjects, there was a certain incidence of mercury resistance. Some people had very, very low mercury resistance in their little Petri plate. And then others had more, and others had more, and up to 100% of the colonies on the plate were mercury resistant. And we also assessed antibiotic resistance by the same means. And we found this curious result, that for, the red bars reflect increasing amounts of mercury. Okay, so what we were first surprised at was that something like 60%, over 60% of the subjects in our study had as much as 100% of their bacteria resistant to mercury as we cultivated it from their fecal samples. Okay, and the other thing that was important was the people who had the lowest level of mercury resistant in their samples also had the highest proportion of no antibiotics at all. They scored for no antibiotic resistances at all. Okay? So we had these opposite kinds of things. If you have no mercury resistance, you're also going to have little or no mercury resistance. You're also going to have very few or no antibiotic resistances in your gut bacteria. However, if you have high levels of mercury, you're very unlikely to have no resistances. Okay, so these countervailing things were kind of puzzling to us because the assumption was that antibiotics were the primary selection for these kinds of plasmids, and the mercury was just on there by accident, maybe because it went through some processing in the sewage and so forth. But we began to look more carefully at that, and we did that in collaboration with Fritz Lorscheider and Murray Vimy. At the time they were at Calgary, and they were the ones who did the initial sheep experiment of installing fillings, and then they later on worked with 4 groups of pairs of monkeys, and we collaborated with them on that. And what we found in experiments like this was we were looking for bacterial changes in monkeys with amalgam fillings, and that was the purpose. And this is an example of 1, 2, 3, and 4 monkeys that we used. And these were actually done earlier. Monkeys F78 and 79 were both treated the same way. They were in the house in the vivarium for a couple of weeks. And this is a week scale, weekly scale. And they were in the house and just, acclimatized to the vivarium in their individual cages. Amalgams were installed. In this case, 16 amalgams, 4 on each side, upper and lower, were installed. And they were left in place for 2 months. Okay, so 8 weeks. And then they were replaced with glass ionomer fillings. Okay? And all during that period of time, we were receiving fecal samples from Fritz and Murray that we plated and studied in our own lab in the way that we had been doing for mercury resistance and antibiotic resistance. And then we also did a couple of more long-term monkeys with them in which the animals acclimatized for a long period of time, and then they had the fillings in for a longer period of time and they were not removed. So I'm going to show you some data from these guys and earlier guys. So here are these guys. How much mercury was there in these animals? And this is a log scale. Okay, so this is in terms of micromoles. So this is down at 0.01, 1/100th of a micromole. And so we go up 1, 2, 3, 4, 5 powers of 10, okay, to 1,000 micromolar, 1 millimolar. And what we have here is the background level in the long-term monkeys down here. So this is the no mercury fillings at all. Then at week 0, the fillings go in. And everything shoots up really high. F78 and 79 shoots up almost 5 orders of magnitude into this peak. And this is the concentration of mercury in the gut during the week. So an average of 2 samples over that week time. Then it gradually goes down. That's the black and red guys. And then when the fillings are removed at the end of the 2 weeks or 2 months period, you get another spike, but it's lower typically by a factor of 10, 5 to 10-fold than the earlier spike. And then it begins to drift back down towards baseline. Monkeys were posted at this point. And you can see that for the animals that received fillings and then kept them, and these guys had 12 fillings each, and they go up not quite as high, and then they come down, they bounce around, but they continue on and drift back down towards a level that is not all the way down towards the original level at all. These were studies that said, well, there's a lot of mercury there, and what's happening to the bacteria? And so we looked at a couple of different categories. There are 4 different categories of bacteria. I'll only show you a couple of them right now. And this is this is either F78 or F79. Before the animals, and this is what we're looking here, is at the percent of bacteria in their feces that are resistant to mercury. So the average resistant to mercury bacteria, and this is gram-negative bacteria in this case, from the gut. And yes, you can see a low level, some 10-15% of resistance here in the adaptation period. But then the fillings go in week 0, and you see a gradual climb in the instance of mercury resistance in these bacterial communities. And then it goes back down again, and so here at about week 7, it has drifted back down to almost to pre-installation levels. And then the fillings are removed, and instead of a gradual increase, as we had seen over here, shoots right up, okay? So that says that the community hadn't really dropped back down to the pre-level because it was pretty close to being ready to shoot right back up again. And then it did drift back down and almost to baseline. And this is a much more rough measure than the mercury measure is. So that the bugs were noticing this. And this was basically the first time that anybody had looked at any ecosystem in the context of amalgam mercury and to see if the mercury was bioavailable, because that was always a remark from the folks who were pro-amalgam, that they said, oh yeah, it comes out, but it's not bioavailable, whatever that might mean. And this clearly showed that it was. As far as the bugs are concerned, this enriched this population, selected for this population of mercury-resistant bacteria, which means the mercury is bioavailable. Okay, well the other part of this was, because of our interest in antibiotic resistance in the first place, was to look and see, under the same kinds of tests, what the percent of antibiotic resistance was. And so at the time, we were doing this without any funding for it, so we only looked at 2 of the antibiotics that we were considering, and we looked at tetracycline, which most of you I'm sure are familiar with, and also ampicillin, which was a common second generation penicillin. And at the beginning of the cooling off period for these monkeys, a couple of the monkeys, and this I think actually covers an earlier group that I'm not talking about. This is a total of, this is the work that appeared in our 93 paper, and I think this covers it covers 4 animals, but 2 of them are the same and 2 of them are different than this animal, the ones that I've mentioned. But there was an animal that came in that had a very high background in both of these. But during the cooling off period, or the adaptation period in the vivarium before the experiment began, that drifted right back down to baseline. These are wild-caught animals, but even wild-caught animals can be impacted by human garbage disposals and stuff like that in the countries from which they come. So at 0 week, there was almost an immediate spike of tetracycline resistance, quite visible spike of tetracycline resistance, and then it drifted down and bounced around a bit. And then then what came up later was the ampicillin resistance. So the tetracycline came up first, and the ampicillin came up slower. At week 5, it was very high, and that corresponds to the peak that I showed you here at week 5. This is the maximum response of mercury resistance, and it corresponds to the maximum appearance of ampicillin resistance as well. And then it bounced back down again, and then after the fillings came out, we didn't see quite as high a burst in antibiotic resistance as we did in mercury. But again, this is a rougher measure. But it certainly spoke to us and said, well, yeah, antibiotic resistance is responding. There's nothing else these animals were being given. There was no antibiotics in their chow or their diet or anything like that. So this is responding to the insertion of amalgam fillings in the mercury in your mouths. So we published several things on that over the years, and from this very first study, actually, we went through that study and looked at the occurrence in all of the bacterial strains, and we looked at some, close to 500 or 600 bacterial strains in this study of many different kinds of bacteria. And we could say very conclusively, if we look at each antibiotic resistance, ampicillin, chloramphenicol, kanamycin, streptomycin, sulfadiazine, and TEC, its prevalence, okay, so this is occurrence, its prevalence was always higher in the mercury resistant strains than it was in the antibiotic resistant strains. And almost all of these, except I think this one, are statistically significant differences. So that's it. If you have mercury resistance in your fecal flora or any other strain of bacteria that's causing you problems, it's very likely the mercury resistance will be, that those strains will also have antibiotic multi-resistance. And that's what you're concerned about. A single antibiotic resistance is not a big deal. This doesn't show it, but all of these strains that have mercury were in fact multi-resistant to antibiotics. And we've done other work with this that I can tell you about if you have questions later. So that was our kind of introduction to the, something's going on here. Later, these are some unpublished studies that we have done in collaboration with Chris Shade, who some of you know, who's a premier mercury analyst and has introduced some methods that make it very much more precise and specific and less expensive to analyze methylmercury and to assess the amount of methylmercury that's present. And that's what we did with him. What's shown here are 2 of these animals from different experiments. This is F78, and this is R87. And this is the time course of exposure. And it looks just like the one that I showed you a few minutes ago. The scale's a little bit different. But this shows a red and a blue line where we Chris reproduced the total mercury number, because total mercury was the only number that we had from the Calgary study. They were not doing methylmercury speciation in that study. So for every sample that we sent to Chris, and he has analyzed some 75% of the total 220 samples of fecal material that we have from these 4 animals, 2 of which are represented here, the red and the blue lines overlay almost perfectly. And that's kind of amazing given the variety of chunks of things and hair and so forth and so on that are actually in a fecal sample from an animal. But they are very, very closely matched throughout the entire course. So we knew that whatever Chris was doing for his total mercury analysis replicated very nicely the total mercury analysis that we'd gotten from Fritz Lorscheider's group when he first generated these samples. Now, This scale over here is in micrograms per gram, also called parts per million. Okay, and again, you can see 1, 2, 3, almost 4 orders of magnitude increase in this animal. And similarly down here, 1, 2, 3, almost 4, let's see, 1, 2, 3, 4, almost 4 orders of magnitude increase here in these animals, in this animal. And good correspondence between the red and the blue marks. One from Quicksilver Company, which is Chris, and one from Calgary, which is Fritz's group. Now the yellow line, which I hope is showing up there, the yellow line is the methyl mercury that Chris measured in the gut samples, in other words, these fecal samples. And say, oh my gosh, it's almost as high as the mercury. The total mercury. Well, no, it's not as high as the total, because this axis over here is in nanograms per gram. So if you're familiar with the parts per million, this is in parts per million, and this is in parts per billion. Okay? So it's a much lower concentration. It's a 1,000-fold lower concentration than the total mercury. This is characteristic of all environmental circumstances in which methyl mercury has been measured, outside, especially in places where there's high mercury concentration. There's always some methyl mercury formation going on. And in every case, the methyl mercury that's formed is 1,000-fold lower. Nonetheless, it's there 24/7, okay, because it was always present as long as mercury was present in the animals. And it drifted downwards in this animal, which is one of the ones that had its fillings removed. But it didn't diminish in the ones that didn't have their fillings removed. You may be wondering about why there's methyl mercury here before the fillings go in, and the answer is that there's methyl mercury in fish chow, and these animals are all eating fish chow as a part of their diet. Oh, thank you. Their diet, animal chow, lab chow, contains around 30% fish meal. And that actually is a subject of some publications in the neurobiology area, because people are concerned about exposure to methyl mercury of rats and mice and stuff involved in neurobehavioral assessments. So this is one way to look at this. And the other way to look at it is something that's really kind of encouraging, and that is with respect to methyl mercury. This is not a timeline over here. This is a plot of methyl mercury versus total mercury. So what we're looking at is, for any sample, what its mercury level is, okay, from 0.001 to 100 over here. How much methyl mercury was there? Does it go up really, the same? This is 1, 2, 3, 4, 5 orders of magnitude, 100,000-fold increase in total mercury. Thank goodness you only get about 2 orders of magnitude increase in methyl mercury. So you're not getting as big a transformation to methyl mercury in this ecosystem as you could if you just based it on mercuric mercury. That's good news, okay? Because if you were going up proportionally to the total mercury, you'd be 5 orders of magnitude up over here too, okay? And that doesn't happen. And it doesn't happen in this ecosystem, and it doesn't happen in natural ecosystems outside, like lakes and streams and other places that are contaminated with inorganic mercury. You always see a lower proportion that goes methylated than you do based on the mercuric concentration there. And the reason for that is, and it's been shown, and actually it's technically shown in our work as well, because in these gut flora that are heavily exposed to mercury, during this period, these guys had lots of mercury-resistant bacteria, which means that they also have this gene. It's called the organomercury lyase, or MERB, and it degrades methyl mercury to Hg2+. It just whips off that methyl group, and you're back to having Hg2+. Okay, so that's a detox gene for the cell, the bacterial cell, that has the effect of converting something that would be very toxic to us to something that's less toxic. Okay? So that said, once it's converted this to mercuric mercury, that mercuric mercury can still go back and become Hg0, because MERB is always a part of a MER operon. It operates when these mercury resistance genes are around. It'll have a MERB gene in there. And so you're ending up through this detoxification for the bugs, going to Hg0, which is going to fly away as monatomic vapor. But it's also the form of mercury that's most readily absorbed across our epithelial lining. And in the lungs, Hg0 going across the lung epithelium has been measured at 80% effective. So if you inhale 100 atoms of mercury vapor, 80% of them are going to go across into circulation across your lung epithelium. And it's assumed, it's not been measured, that any epithelial tissue would be about the same. So the bugs are detoxifying the environment for themselves. Fortunately, one of the things they can do makes a little bit less methyl mercury than could be there. But the end result of the bug's process, as I said, it's giving you another chance at that mercury atom. Just put it back out into circulation, get that thing oxidized, and see what it attaches to. Okay, so that's kind of good news, but it's not the best news that we'd like to have. We need some friends, and the bugs are not always our friends. So the studies in living animals told us that amalgam-derived mercury is bioavailable. Mercury-resistant bacteria are also multiply antibiotic resistant. So if you have mercury fillings, once you go off that antibiotic, it doesn't matter. Those antibiotic-resistant bacteria are going to continually be selected by the mercury coming down through your GI tract. And amalgam-derived mercury can be methylated by microorganisms in the bowel 24/7. Okay? All right, now, knowing this, we really got to thinking about, well, just why is mercury toxic? I mean, there's some sort of paradoxical things, some of which I'll mention here in a minute. And so just in case I get rushed at the end, which, what have I got? We got half an hour or 20 minutes or 29 minutes. Okay, that's good. Thank you for the reminder. Okay, this is the cellular and biochemical studies. And we've used bacteria again because they're malleable. And we're going in to look at all the proteins in the cell at once. And so you want to start off a project like this with a cell that has not too many proteins. If you're developing a method specifically to look at which proteins are the ones that get hit with mercury, which ones are the most vulnerable. Okay? So this is where we were at this point, and this is, I should remind myself here, since I think I'm sort of doing good on time, and I hope I won't get too bogged down. Are there questions on what I had to say that you'd like to ask me about just quickly that will help you go forward with me? We can do other more general questions at the discussion group later on, but if there are things that, oh, wait a minute, wait, don't go any farther kind of questions from anybody. This is an invitation for what you would call a stupid question, and I would call, really, that's an insightful question. Yes, we're so happens all the time. Okay, no little waves from anybody out there? All right. Okay, so moving along to this next section, we were really, sort of torn with the business of, well, now we know that there's all this mercury coming out of fillings. And how can it be, as I mentioned earlier, and actually, Mark covered very nicely in his talk about, there's a lot of mercury here. Why aren't we dead, sort of thing. And part of it does have to do with some buffering that I'll talk about later on that we have evolved to have. But we really wanted to get at this. And the other thing that we were concerned about is that there are many scientists who work on all kinds of proteins that are involved in cellular biology and are completely unaware that there's this much mercury bathing the ecosystems in which those proteins and cells normally exist inside of a human. And so we thought one of the things we could do is go in and look at the proteins that are the most vulnerable and begin to let other scientists who are building their whole careers on this, that, or the other protein. By the way, if you're trying to develop a drug or if you're trying to develop a diagnosis, you want to take into account the fact that your particular protein might, in fact, also be impacted by something you're not aware of. And we also wanted to work out a diagnostic method that would allow us to determine whether or not a given protein in any given human that might be vulnerable is, in fact, impacted. And this is the general area called biomarkers. Okay? So this is a quick précis of what I'm just about to tell you. Okay? About modification of proteins in the cell by mercury in every major metabolic pathway. Oops. but I fixed that. And how it's especially damaging to proteins that are involved in oxygen defense, and how such a subset of these E. coli proteins are evolutionarily conserved in humans. So let's go to my reasons for saying that I can tell you that. People study mercury a lot. We've heard about NIHS, and we've heard about EPA and others, and literally tens of millions of dollars a year are spent on what is really a geochemical cycle of mercury. We've heard about the carbon cycle with photosynthesis and stuff that we're concerned about with global warming, and you may have heard about other cycles like the sulfur cycle and so on. Well, there is a mercury cycle because mercury can exist in so many different chemical forms, and there are both biotic, which I'm talking about right now, as well as abiotic, which means things like sunlight and ozone and so forth generated chemically that can mediate all of these changes among these several forms of mercury, which Mark mentioned earlier, the organic form and the two inorganic forms, mercury 0 and mercury 2+. So the data, and actually this is a little bit behind, the global atmospheric burden of mercury is in the neighborhood of 1,000 to 2,000 metric tons. And it hangs in there for about a year at a time as it circulates by rain and stuff back into the water and onto the land. And, U.S. fossil fuels contribute about 46 metric tons per year to that burden. There is also local contamination from industries, including the chlor-alkali process that Mark mentioned. There used to be isotope processes, and now DOE's big problem is the legacy of those isotope processes that were Cold War legacy. Batteries used to have mercury in them, and some of them still do in dental offices. Okay, so some of these things are regulated. Chlor-alkali isotope processes are basically gone, and they are regulated severely. The residue is regulated severely in batteries. And, of course, fossil fuels are regulated with the expenditure of many millions of dollars, both in regulation as well as in devices for removing the mercury from the stacks. The only thing that isn't regulated is dental offices and the dental process. So, this is our little circle down here, and when I made this slide, it was a few years ago, there's 30 metric tons of mercury are used in dentistry every year. And the average amalgam at the time, the number I had was about 10, so there, if we just use a ballpark napkin type calculation of maybe 5 grams of mercury per person, at the time the U.S. population was around 30 million, and 80% were said to have some amount of mercury fillings. And I know Mark is cringing at my calculations here because it's a worst case scenario, but that would be 1,200 million grams in U.S. mouths, or roughly 600 metric tons of mercury walking around in people's heads just in North America. And they're walking around. Okay? So, here's the paradox. How can this stuff be toxic? Okay? It's, isn't it kind of unbelievable if that were toxic? That this could be happening? And this was the conundrum that we faced ourselves as scientists. Well, what's going on here? So this was just meant to refer to the people that, these people that work in the plant could be affected by that. People having dinner and eating fish could be affected by that, but each one of them also has a hot spot for mercury in their mouths. Okay, so let's think quickly about mercury intoxication. Why are organo and inorganic mercury toxic? Don't be frightened by this. This is just an equilibrium, a reaction that shows if this ion of mercury interacts with this other kind of ion, it's going to form this complex. And then if it gets another kind of, or it starts out with 2, it's going to get a neutral complex. And that's just the statements for these numbers down here. The affinity constant for this one is if you only have one associating ion, and then the affinity for this one is if you have 2. So most of the time mercury takes 2 ligands. And here's mercury acetate, which has a very weak association. Mercury chloride has a stronger association. Moving down the halide portion of the periodic table, mercury bromide is even higher association. And mercury iodide is very, very strongly, in fact, complexed to mercury and iodine. But the strongest of all is to go over here and think about sulfur, okay? So if you have, in this case, we're talking about not an inorganic sulfur, which would be sulfide, HGS, S is 2 minus, so that you just need one S with one mercury. It's called cinnabar. It's one of the common ores of mercury. It's a deep red color. But the association constant here is 15 to 17 orders of, or no, more than that, close to 17 orders of magnitude greater for any sulfur compound. These things are referred to as almost covalent. You think of covalent, that means like carbon bonds and stuff. Yes. Sulfur is considered in terms of its stability almost equivalent to a covalent bond when it binds to mercury. Okay? Selenium is even stronger. Okay? And we'll come back to this in a minute. The other point I want to make to you is the chelation effect also makes this stronger. Because if you have one agent that has 2 sulfurs on it in the same backbone, it's called a dithiol. And an example of that is, a common example is the compound used in chemistry called dithiothriotol, which is a sugar that has 2 sulfur groups on it. It's an artificial molecule. That makes a very strong, and it's much stronger even than just 2 monothiles binding on the sides of mercury by themselves. And the reason for that is, what I'm showing you here is thermodynamics. This is the thermodynamic stability of mercury compounds. Okay? However, monothile adducts can rapidly exchange. They can flitter around all over the place. Okay? So if you go in with this kind of an HGSR complex, which is this one, okay, that has association constants of 35 to 40, and you throw in just another monothiol, like glutathione, for example, or a protein that has a single thiol on it, it'll flicker on and off and on and off as long as there are thiol groups making it possible for it to exchange. Okay, so the reassociation of monothiles is very rapid. What's tricky and dangerous is when you have a protein represented by this scrawl here, a protein folded chain, and the protein chain has 2 cysteines on it, then it can attack mercury with the chelation effect. And it's much harder. In other words, it's 100,000 times harder to get the mercury off that protein, okay, with the usual kinds of ligands that the cell provides. You're probably familiar with glutathione. You may be familiar with an acetylcysteine or cysteine as reagents for detoxification of mercury and so forth. All of those, including DMSA and whatever the other ones, they are all monothiles. They can't get mercury off of this in real time. Okay? So if you've got mercury cotched by a protein with 2 cysteines, and it may be as many as 3 or 4 cysteines, it is not going to come off in real time with a monothiol. That's just chemistry. Okay? Okay, so what did we want to do? I'm going to introduce you in this slide to proteomics, which is looking at all the proteins in the cell at one time using mass spectrometry. And you know we have got genes sequenced, and we know the length of the protein, all of its amino acids, and we can make a prediction about, well, if we treat this protein with trypsin, which cleaves at 2 particular amino acids, either arginine or lysine, we'll get some fragments. Because we know the sequence, and we know where the lysines and arginines occurs, we'll get a set of fragments. Okay? Big ones and little ones. And if we throw these into the mass spectrometer, 1, 2, 3, 4, 5, 6, 7, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I'm not quite sure there's that many there. But we will get fragments that represent each one of the subfragments, arginine-lysine fragments in this peptide. So we can do an in silico prediction of what we should see in the mass spec. Then we can take the real protein, okay, and we can use a protease like trypsin and chop it up into little fragments and throw it into the mass spec, and it'll tell us, okay, all right, those 2 are there. This one's not as abundant, but it's different peptides fly differently, as we say in the mass spec. We've got a couple there. We've got these 2 guys. We got this singleton right here. We got these 3 guys. And you can make a match and see for yourself that, oh yes, we got this many peptides out of this protein. You can use, this is like a fingerprint. You can fingerprint proteins by mass spectrometry. You can actually sequence them, so you can even know exactly what the peptide sequences are in the mass spec by using the mass spec. So what we wanted to do was use mercury, which is which is, well, let me say, Mercury has a bunch of isotopes. I'm not going to try to go through all this, but Mercury has several stable isotopes. And by itself, though, it has a mass of 200. So the average mass of Mercury is right around 200. And I'm going to skip this. I'll come back to it if somebody wants some more detail. But suffice it to say that you can use this mass spec proteomics method, or we have established that you can do that, to look at the mercury-modified proteins in the cell. Okay? Because you're going to have, if you added 200 additional mass units, 200 Daltons, you're going to have an extra peak that's not represented down here in your theoretical protein sequence. Okay? I mean, there will be some of that in the cell, but to the degree that it's modified by mercury, more of it will occur in a form that gives you this extra peptide peak, because that's one of those little peaks, okay? Maybe certainly a smaller peak, some smaller peak than these last ones here, was modified by mercury, like there. I guess I put a red one on that. And that's what's showing up here as an extra peak in the mass spectrum. Okay? So you've changed the fingerprint, and you can identify those things actually with computers that are used for doing this. So what we did with E. coli was to expose the cells to mercury. And again, I'm not going to go through this in detail, but I will just tell you a couple of things. You grow the cells, you expose them to mercury, you harvest the cells in a centrifuge, and you send them off to the guys at the Pacific Northwest Laboratory in Richmond, Washington, and they do the mass spectrometry, which I'll tell you about in a moment. We also do a lot of benchmarking assays at Georgia to find out, to be sure, well, first of all, to find out what is going on in terms of the bulk biochemistry of these cells. And I'll tell you one thing about one item that we found, and also to make sure that they are standardized, because we grow them the same way, and we want to be sure that they turn out the same way each time before they go out to be treated in this fairly expensive process of mass spectrometry. But it's growing less expensive. Okay, so one of the things that we found in looking at the bulk protein products, the bulk cellular results of exposure to mercury or mercurials, is that mercury exposure increases intracellular free iron. Now, you may know, as you're, I think, concerned about oxygen-related phenomenon like reactive oxygen species that can cause damage in the cell, and that an agent of carrying out that kind of chemistry is iron in the cell. And so the cell goes to a long way. Bacteria is one of those. We go to a long way to make sure that iron is either fully reduced in the cell or sequestered in some way, that it can't just be like flitting around and getting oxidized and doing damage. Okay, so the Fenton reaction is what it's called, and its agent is basically free iron. And so we see a fold increase. So this is fold increase in free iron compared to unexposed cells, and we measure this with a device called the electron spin resonance or electron paramagnetic resonance. And there's a certain background level of free iron in the cell, and that's marked as 1 here. If you add 4 millimolar hydrogen peroxide, it goes up a squeak. The cell has enzymes to take care of hydrogen peroxide. If you use phenylmercury at 100 times less than that, okay, it goes up a bit more, okay. With merthiolate at 40 times more or less than hydrogen peroxide, it hardly goes up at all. But both of these compounds have some difficulty getting into the cell. However, mercuric chloride at even 16 micromolar almost doubles the amount of free iron. And if you go up to 80 micromolar, still well below the standard oxidizing agent. If you go up that far, you can actually see at the limits of some of our variation here, you can see almost 80% of the cell iron is free. And the reason for that is that there's a class of proteins called iron-sulfur proteins in the cell that are very ancient and have been around for a long time, managing oxidative responses and managing electrons for the cell. And these proteins are direct targets of mercury because they're full of sulfurs. And ordinarily they'd be holding on to an iron. But mercury can come along and displace the iron and hold on to those sulfurs so that the cell is left with the protein is left with a mercury in its mouth, and its iron is out there floating around, coming to harm and doing damage to the cells. So by participating in Fenton and Haber-Weiss reactions that form what are called ROS, so reactive oxygen species. Actually, that mercury does release reactive oxygen species or provoke that has been known for a long time. In particular, in eukaryotic cell biology, it's known to be an aspect of mercury's damage to mitochondrion. Well, the mitochondrion used to be a bacteria a long time ago, as you may know. The endosymbiotic, it's not a theory, fact, of the evolution of our cells is that they were formed at some ancient time by joining two different kinds of bacterial, two different kinds of microbes. And so the enzymes that are in our mitochondria are essentially the bacterial versions of those enzymes. And many of the iron sulfur proteins in our cells have very ancient lineage in bacteria. And this makes clear that the cells are very much damaged by that. This is a quick summary of the proteins that we saw that were damaged. In other words, that had adducts of mercury. They were tagged with mercury or organomercury in our proteome experiments. So the blue right here is the E. coli wild-type protein distribution by function. So if E. coli has about 4,300 proteins that it makes. Doesn't make them all the time. But if you look at the different percentages of E. coli's proteins that are in different functional groups, about 4% are involved in gene regulation, about 4% are involved in the cell envelope, making the cell envelope, about 7% are involved in transport, and about 8.5% to 9% are involved in energy metabolism. And that has to do with making ATP, for example. I'm not going to go through all of these. You can ask me about them later if you like. But the things I want to highlight here, energy metabolism is a real biggie. Protein synthesis is a real biggie. You've got to make proteins if you want to divide. And amino acid biosynthesis, because you've got to make amino acids if you want to make proteins. So these are some spikes that turn up here. And the reason I'm highlighting them is of this 4,300 proteins of E. coli, we found about 329 proteins that were especially vulnerable to mercury. They would bind mercury to their cysteines very tightly, very stably. We could see it regularly in experiment after experiment. And we have done a total of probably 8 partially complete experiments, of which 3 were fully complete experiments, fairly long and complicated experiments. But these are reproducibly observed as these 329 proteins, 309 proteins are reproducibly observed, and that's what these red guys are. And I'm going to point out primarily the ones that give us the highest spikes and the ones that seem to exceed in their vulnerability the average distribution of things in the E. coli genome. So just quickly, these are going to be examples of proteins with cysteines that are modified 80 to 100%. So this up here this is the amino acid biosynthesis guys, and some examples of that are tryptophan synthetase. That's a very important protein. Tryptophan is synthesized by bacteria. Ornithine, which is involved in arginine synthesis. Glutamates and other amino acids, very important. And the hydropicolinic, which is involved in lysine biosynthesis. All of those are important and essential amino acids for us. This is the energy metabolism bunch over here. Okay, ATPase, that's the thing that makes ATP. It has multiple subunits. 3 of those subunits are hit with mercury in an extensive way. Okay, glyceraldehyde 3-phosphate dehydrogenase is an important part of glycolysis, which is non-oxidative metabolism. NADPH, ubiquinone, which in the membranes, NADPH or NADH is exchanging reducing equivalents with ubiquinone. If it doesn't happen, the membrane can't function. And succinic dehydrogenase. Both of these are very important in the electron transport pathway itself. And of course, ATP synthase uses the electron transport pathway in order to make energy. And all of those guys are taken out. And then protein synthesis. I didn't bring a drawing of that. A ribosome is the particle, the engine of protein synthesis in the cell. Ribosomes have 2 particles, a big one and a little one, In bacteria, they are called, the little one's called 30S and the big one is called 50S. Between the two of them, they have over 50 proteins that are stuck into RNA. So they are mostly RNA organelles, but they have proteins stuck in all over them. And for both the 30S and the 50S, there are 7 proteins in each subunit that are heavily modified with mercury. And they're not doing their job if they're heavily modified with mercury. tRNA synthetases, these are the little enzymes that go around, grab a tRNA, grab an amino acid, put it together, send it to the ribosome, make a protein. They're the basis of coding. You've got to get that right, okay? You can't make mistakes. You're the dictionary master. And we have, I think, 6 or 7 of these that are damaged. And the ribosome has little acolytes, little proteins that come and go that make it do its thing. Starting to make proteins, continuing to make proteins, and then stopping the protein. And these are called factors. Initiation factors, elongation factors, and termination factors. And we've got several of each down here that are so extensively modified that they would not be functioning under the circumstances of mercury exposure that we used here, which was a toxic dose. We did use an acute toxic dose in these studies because we wanted to be sure to see something, but it's still in the micromolar range. It's not in the millimolar range, which, as I pointed out, you can easily get millimolar range just in the GI tract. Okay, and we've pointed out here some selected human mitochondrial homologs of E. coli proteins. We have a succinic oxidase ourselves. The cysteine sits in a peptide in that protein that is identical to the peptide that it sits in E. coli. In fact, that's true of all of these. Every one of these, we've picked out this subset of, I think, 2, 4, 6 of them, that their peptide is identical to the peptide in E. coli. That's strongly evolved. Right. Okay, and these are all, I mean, all of these are involved, or 3 of these are involved in multiple disease associations. So succinic dehydrogenase has multiple disease association. I mean, these are in genetic screens as showing up as polymorphisms in genetic screens in multiple different diseases. Tyrosine, tRNA synthetases. 2 of them are not do not show up as screens in diseases, but it's possible that they are so valuable that you can't even get born, and so you're not going to be screened as having a mutant in that respect. And I'm not going to try to show this one, but just to mention out that the succinic oxidase here is one that's been named in a recent paper on autism, mitochondrial dysfunction in autism. The enzymes that they measured that were particularly dysfunctional in their cohort, succinic oxidase was one of them, and adenosine triphosphatase, ATPase. So in summary, I just realized I left something out here, and I want to go back very quickly and show this. I should go to, but I think I can get there quickly. I didn't point out in this slide with the bacteria this point. You may have noticed it, okay? But this is 1 part per million right here. That's the fish advisory level. In the monkey gut, when these fillings are in place, you will notice that almost all of the line here is above 1 part per million. Here and here. And that's true of our other monkeys as well. So for the duration of the time that the mercury was there, the gut contents were well above, at least 10-fold, in some cases almost 100-fold higher gut contents than would have been allowed by fish in the store. The monkey, there's no way the monkey could have eaten fish that would have gotten that gut concentration that high. Okay, so sorry about that, and I'm just about at the end of everything else. Ding, ding, ding. Yes, human homologs, mitochondrial dysfunction. So this summary is this, brief exposure to inorganic or organic involves modifications in every major metabolic pathway. Prominent include energy generation, protein translation, amino acid biosynthesis, and redox proteins are especially vulnerable, and approximately 10% of the highly vulnerable proteins in E. coli had homologs in humans. So, what's next? I'm going to go beyond bacteria to work in human models and animal systems. The animal systems that we have the closest association with are mouse and nematodes. You may know that C. elegans, which is a nematode, is an important metazoan model. Before we go to those peptide hormones, humans have I think it's about 60 peptide hormones, 50, 60 peptide hormones. 26 of those have cysteines, from 1 to 13 cysteines. Insulin is a good example. Out of this small 2-chain polypeptide, it's 2 chains, insulin has 4 cysteines, and those are very essential in correctly folding the protein and making it take its stable functional shape. Nobody's ever asked if mercury interferes with that. So we're going to look at peptide hormones. We're also going to look at selenoproteins, and the reason for that, I think I put back in here, is how strong the selenium association is. Very, very high association. Humans have 25 selenocysteine-containing proteins that we depend upon, and they're involved in our redox defense. They're the proteins that can come to the rescue of a sulfur protein that's been oxidized by oxygen. So selenium is the really tough guy in terms of being able to peel oxygen off of a damaged sulfur or cysteine, and that's done with selenocysteine. It's also involved in thyroid function. In fact, thyroid hormone is made by a selenoprotein. And thyroid, I don't need to tell you guys, is practically at ground zero with respect to mercury exposure. And we're also, well, the reasons we're doing this is to come up with diagnostic biomarkers where we could go into a tissue system or the blood or other kinds of bodily fluids and use an antibody pull-down technique to just pull out a given protein and then take just that protein and look at it by mass spectrometry. And you think, oh, you just told us those were big, expensive machines. No, you're not going to have one of these in your office. But they are getting less expensive, okay? But this would be something that, you know, the sons and daughters of Quest and other kinds of companies would be able to use fairly readily. And in fact, I'm sure many of them are already using mass spectrometry. That would allow us to help in resolving drug trials and the genetic basis of disease. Many people are very frustrated that the genome didn't tell them that this gene causes this disease and that we can make this drug to make this disease go away. That's a huge disappointment for people working and a huge frustration for people funding genomic research. And sure, there are many levels of things going on. There's modification, there's epigenetics and modification of the genome, but there's also the problem, as I said at the outset, that many of the people in clinical trials might not ought to be in a clinical trial if they're heavily poisoned with mercury, because there's going to be a severe compromise in many of the proteins that might be involved in what they're trying to cure by the fact that the mercury's there. And these could provide diagnostic tools to help clarify clinical trials for other drugs. And it's also, mercury's a great model. Most toxins are electrophiles, and bisphenol is an example, and it's a compound that's going in there and wants your electrons, and it's going to get them. And it might even attach to you, you know, just to get your electrons. All right? And that's what mercury does, and we don't know to what degree other kinds of electrophiles also do that. And so this business of searching for these modified proteins by this particular mass spec technology, mercury's a good model for other electrophile toxins. So, and then what kind of therapy can we come up with for people who have been poisoned by mercury? We have to keep in mind that whatever we come up with remembers that chemistry that I talked about, okay, that you're going to get the stuff with a bis chelate and maybe a trigonal chelate. And also, you have to take into consideration that you've got to keep it from going into the enterohepatic cycle. So whatever you dose with has to be something that really can't itself go back into circulation. And it might be a two-pronged approach. So anyway, we can talk more about those things. I want to acknowledge quickly the collaborators, particularly on the mercury work, my own group, Stephen LeBoy and Lynn Olive, and Taka Sasaki doesn't work on this particular part. My colleague at UCSF, Sue Miller, and her postdoc, Ben Polacco. The group at Pacific Northwest Lab, proteomics, Mary Lippin and Sam and Erica Zink. And then we also do some computational chemistry and ecology and evolution with other people. And as I mentioned on the first slide, we've been funded by the Department of Interest, and thank you for your interest.